Good morning, everybody. Happy holidays, and welcome to our final Medical Grand Rounds of 2021. We have, as usual, and particularly today, really great speaker. We have some COVID updates. We wanted to give a little bit more COVID updates, given this is the last Grand Rounds of the year. I'm going to turn it over to Dr. Ben Pinsky, who's going to provide us updates next on the variants. Thanks for, as always, and again, for being with us throughout the year, Dr. Pinsky.
Thanks, Errol. A very brief update on our surveillance for Omicron. We've now identified 5 Omicron cases in specimens submitted to the Stanford Clinical Virology Laboratory. 3 of those cases have been confirmed by whole genome sequencing. These were from a single household. 2 of the individuals were vaccinated but not boosted. The other, there's no information on. All had mild symptoms. We yesterday identified 2 probable cases by mutation-specific PCR. Sequencing is pending. These, we're using a new assay that identifies specifically Omicron that has this deletion, specific deletion insertion. One of these individuals was boosted, also had mild disease. I should say all the cases. No international travel. One of the cases traveled from Florida and apparently was sick on the airplane, so that's unfortunate. This is what we have so far. A vast majority of our cases remain Delta, but of course throughout the world and the rest of the United States, there are very rapidly increasing numbers of cases of Omicron, so we will be ready to identify these when they arrive. So thank you for the update, and I'm happy to take questions in the Q&A.
I'm next going to turn it over as we end the year of Medical Grand Rounds to Dr. Harrington.
Thanks, Errol. I just wanted to take a moment and to thank our Medical Grand Rounds audience for joining us every week. You've really, I think, shown us one of the positive things that can come out of the pandemic, and that is the more rapid dissemination of information. And many hundreds of you have joined us every week, and we're appreciative of that. And we hope that we've kept you both informed and perhaps even a little bit entertained. As I look at the screen, I want to thank all of the panelists who show up week after week to answer questions in the Q&A, to have chat conversations with members of the audience. It's really been an important addition to what we've done or tried to do every week, as we want to share with you updated information about the pandemic, but also about broader issues affecting our Department of Medicine community here at Stanford. A really special shout out to 3 people. Brian, for coming every week to give us the occupational health update. Thank you, Brian. And we very much appreciate it. Ben, for showing up every week and sharing with us the information from the pathology lab and the really heroic work that your team is doing. So thank you, Ben. And I'll save my special thanks for you, Errol, that Medical Grand Rounds has really succeeded because of you. And so thank you. And my final wish is that you all take some time to rest, to reflect, and come back re-energized in 2022, because we all have a lot to do. So with that, happy holidays. Let me turn it back over to you, Errol, and I really look forward to today's lecture. Thank you.
Thank you, Dr. Harrington. And I might need to say it, but you've been leading us through this before the pandemic, through the pandemic, and after it. And thank you so much for everything you've done. And just to add, I appreciate it. And I hope everybody's having a wonderful holidays moving forward. I want to turn over next to Dr. Halotny, who's going to share with us a little bit about this special lecture we have today in the lectureship and what it's about. Dr. Halotny, thank you for being with us today. I'll turn it over to you. You're currently muted.
Let's try that. How's that?
Perfect.
Okay. Good morning, everyone. I want to welcome everyone to this special Medical Grand Rounds event, which today is sponsoring the 24th Thomas C. Merigan Lectureship. Before Dr. Relman introduces our guest speaker, I want to take a moment to tell you all a bit about Tom Merigan and this lectureship. Tom became an assistant professor at Stanford in 1963, subsequently heading up the Division of Infectious Diseases and founding the Diagnostic Virology Laboratory at Stanford. After becoming the first member to hold the Becker Chair in Medicine in 1980, Tom became the principal investigator of the NIH-funded AIDS Clinical Trial Unit here at Stanford and director of the Center for AIDS Research at Stanford in 1988. After Tom stepped down as head of the Division of Infectious Diseases and Geographic Medicine here, a group of his friends and former fellows like me established the Thomas C. Merigan Lectureship in his honor. They created an endowment recognizing him for more than 4 decades of research, teaching, and patient care, and for his lifelong commitment to understanding the nature of the host's immune defenses against viral diseases and designing methods for their control. Dr. Merigan and his fellows were pioneers in the use of interferon and other antiviral agents, developing viral diagnostic assays, and in understanding how the immune system controls virus infections in humans. Through our conversations during the COVID-19 pandemic, I knew Tom continued to be interested and engaged in understanding this new viral illness. To honor Dr. Merigan, the lectureship seeks to bring other scholars to Stanford who, like Dr. Merigan, have devoted their professional life in understanding the interface between infectious diseases, the host, and their environment. Jonas Salk gave the first Merigan lecture here in 1994, and I'm sure you'll agree that our speaker today certainly embodies all these attributes, and I can't think of a better person to carry on the tradition of the Merigan lectureship. Dr. Relman.
Thanks, Mark. It's my great pleasure to introduce Dr. Pardis Sabeti as the 24th Thomas Merigan Lecturer. Dr. Sabeti is a professor in the Center for Systems Biology and the Department of Organismic and Evolutionary Biology at Harvard University. She's a professor of immunology and infectious diseases at the Harvard T.H. Chan School of Public Health, an investigator of the Howard Hughes Medical Institute, and an institute member of the Broad Institute of MIT and Harvard. Pardis has had a remarkable career over the relatively few years since finishing her formal training in 2006. Her work is a creative blend of genomics, evolutionary biology, information theory, virology, and public health, focused in part on developing comprehensive approaches for detecting, tracking, containing, and treating deadly viral diseases. Among her many contributions are new technologies that enable rapid field deployable tests for a wide array of viruses, tools for recognizing adaptive changes in the human genome, insights into the real-time evolution of viral pathogens, and experiential learning modules and mobile apps that facilitate responses to disease outbreaks. She is a passionate advocate and leader in STEM education, data sharing, and in the empowerment of those with few resources so that they might actively participate at the front lines of public health response. Dr. Sabeti is a graduate of MIT, a former Rhodes Scholar with a doctorate in biological anthropology from Oxford, and a summa cum laude graduate of Harvard Medical School. Among her many honors, she was a Time Magazine Person of the Year in 2014 for her work on Ebola, a recipient of the Richard Lounsbury Award from the US and French National Academies of Science in 2017, and was elected to the US National Academy of Medicine in 2020. Finally, I will simply suggest that if any of you are in need of a brief break and some music, you might enjoy the results of a Google search for a rock band called 1,000 Days. But please wait to do so until after 9 o'clock Pacific. Pardis, we are grateful to you for joining us here today.
Thank you, David, for that lovely introduction and for the honor to get to be here today for the Thomas Merigan Jr. lecture. So it's a real honor, and the more I read about Dr. Merigan, the more I was excited by everything that he's done and contributed over 4 decades. Humbled to get an opportunity to speak on his, in his honor. So thanks so much, and great to be here with you in the last days of 2021. I'm sure, like everyone else, you're feeling that heat where every single thing that had to happen in 2021, people want to get done this week. On top of Omicron, it's bananas. But here we are. I'm actually showered, but that might be about all you have for me, but I'm excited to dig in. So let's see, here we go. All right. My presentation, okay. So I'm going to be speaking today about the work that my lab has been doing over the last decade or 2 decades, of kind of formation, depending on how you count it. But ultimately, I think a long time ago, I became really fascinated with infectious disease from an evolutionary standpoint. They are, incredible. They are both major drivers of our evolution, but also evolved themselves. And understanding also kind of being, cutting my teeth in the genomic age as it developed. And so I've really thought for a long time about this idea of how we can respond to pandemics in this genomic and information age that we're part of. I just want to, before I begin, just note that I do have conflicts of interest that you should take into account as I speak about some of the touch upon these topics. Specifically, I'm the co-founder, consultant, and shareholder in a company called Sherlock Biosciences that uses CRISPR-based and synthetic biology to develop diagnostics for infectious disease. I'm also a board member and shareholder of the Danaher Corporation that owns a number of companies, but in particular, I want to note Cepheid and IDT as they relate to diagnostic work. So I'm going to take us back a bit. I know when I started with COVID, I really stopped talking about Ebola and didn't want to become known as just the Ebola person when this global pandemic was going on. But then actually over time, I realized that it was good to dust off these old slides and bring them back. And mostly because of this idea that, history doesn't repeat, but it rhymes, and viral infections aren't all the same, but they do have a whole lot in common. And the good thing about that is if we think about it, that how we, react to one virus is informative for the future. So in March of 2014, you may recall, you may not, the World Cup was coming up and there was a lot of action in the Middle East, but some of us were paying a lot of attention when in March of 2014, Ebola was declared in West Africa. And this is of course of note because Ebola hadn't really been seen in West Africa before, even known to be there. There was a large outbreak, about 60 individuals in Guinea who were identified to have Ebola. But when they, actually went back and did epidemiological investigations, this was not the start of it. It had been going on for some time. In fact, the epidemiological investigations traced this back to late 2013, around 4 months earlier, in a remote village in Guinea where they believe the index case, the primary case, was a 18-month-old boy who had been playing in a tree inhabited by bats, and it had been disturbed. He came down with this mystery illness and passed away, unfortunately, but not before also spreading that to his loved ones and to the village. And by the time the outbreak was declared, it was in districts throughout Guinea. And by that was in that late March. By April, it was in Liberia. This area between Sierra Leone, Guinea, and Liberia are very interconnected. So it's not surprising, but just the breadth of where it had reached, was remarkable. And now it's starting to get to, other areas that might be more populated. And then in May, when it came into Sierra Leone, it came like a tidal wave. And if you recall at that time, those early days, it had already escalated to the point that it was, one of the largest, if not the largest, outbreak of Ebola we'd ever seen. But in that point, when it came into Sierra Leone, the trajectory changed. It suddenly took off. And a lot of people, kind of pointed to demographic effects, that Sierra Leone had a lot of highways and big cities and that there was something else going on there. People pointed to the fact that the MSF and WHO and other first-line responders were tapped out in Guinea and Liberia, and so just weren't able to respond. And it was us researchers that were trying to manage with very limited resources. But to us, it seemed like something was different. And my group got a frontline view to it because we were not outbreak researchers. We were researchers of viral, sort of host and viral determinants of viral hemorrhagic disease. We'd been working on Ebola, but primarily we've been working on another disease called Lassa virus at the Kenema Government Hospital in Sierra Leone. And we had, in order to do this research with our colleagues at Tulane and now at Scripps and elsewhere, we had set up the ability to do diagnostics for Lassa virus on site, both antigen and antibody-based as well as molecular tests. And so when we got that alert, where I was paying attention to those alerts, we immediately sent a team, as we do, to go out and help establish diagnostics for Ebola and for the circulating lineage of Ebola that we were seeing. So to the right are, in sort of starting with khaki, is Christian Anderson and Stephen Geyer, then on my team. Christian is now at Scripps. But they flew out and helped set up diagnostics right away. We already had those diagnostics tools in place and just brought the guides to test for Ebola. And in the blue shirt in the middle is Dr. Humar Khan, who was the head of the Lassa fever award and arguably the world expert on hemorrhagic fever, having seen more patients than likely anybody else on the planet. And we were working together, our teams have been working together for about 7 years at that point on these diseases. And so we had already established this ability to sequence virus at that point, mostly from Lassa and other co-circulating viruses, but we also were able to sequence and to do so with a very limited footprint. We essentially took clinical excess samples from diagnostics, put them in a tube, deactivated them, and sent them to the United States, where we did sequencing at that point. And so when they were doing surveillance in March and in April for Ebola, hadn't seen any cases. And then in late May, a woman came in who was having a miscarriage, tested negative for Lassa, and they pulled out the Ebola test and tested positive. And came in not just as an individual case, unfortunately, but as a cluster of cases connected to a funeral. And before we knew it, we were sitting on a massive outbreak of Ebola. The samples were sent to us in Massachusetts, and we immediately started sequencing. In a matter of weeks, had hundreds of 99 genomes from patient samples that we had generated, and we're finding hundreds of mutations that were popping up. And this is not new at that point, right? We all know that viruses mutate, but that data actually was pretty alarming to have that much data in that short of a time that was made possible by this genomic revolution. And by the fact that we were there at the right place at the right time to catch it, we were able to see these hundreds of mutations. And both to alert the Ministry of Health that this was indeed Ebola, and to inform the world about how the virus was changing. And one of the things we did then is we shared that data immediately in real time. And when we did, it was funny reaction. Like we had colleagues on the front lines, and I cut my teeth in human genetics. It had the Fort Lauderdale principles, a lot about data sharing. And so we immediately released the data to say, world, this is what's going on, before even beginning to put pen to paper to put something together on a paper. And we immediately, Andrew Rambeau and others immediately reached out to us and said, did you mean to share this data? Did you, what's going on here? Where's the paper? And we're like, we don't have it. We just need to work and get to it. And it was tremendous to get to work collaboratively with lots of colleagues to analyze this data. And it's so important to share that data in real time, as we all see now, because it can inform diagnostics, vaccines, and therapies. It can help us identify variation across the genome and begin to see these different lineages that are popping up and where they're popping up, to track and characterize outbreak transmission. And in this case, essentially, while co-analyzing with 3 genomes that were released from Guinea, previously we were able to see that this was all kind of connected, that this was not multiple entries from the environment over time. This was a single likely transmission into the human population, and from there was transmitting from human to human very quickly. And we began to investigate functional mutations. We found a mutation that really caught our attention, that popped up just as that trajectory changed, just as cases really took off, of an alanine to valine change in Ebola's glycoprotein. And this is unusual, and my colleague Jeremy Lubin, who I've been working with since that time, and I began to sort of collaborate and think about this, and work together to show that this single mutation in the glycoprotein increased infectivity of the virus. And then working with other colleagues like Jonathan Ball, we're able to show that in a number of other mammals that this decreased the infectivity. So in many other mammals, including bats, it looked like that singular mutation decreased infectivity. So kind of to us, what was clear is this virus is having more and more opportunities to transmit from human to human. It appeared to be getting better at doing so. And this kind of information is really important. This idea of human to human transmission, these kind of key concepts, it shows you what genomics can tell you. That understanding, you may not recall, but early in the outbreak, most of the messaging was about don't touch bats, don't touch monkeys, don't touch fruit and vegetables that were touched by those animals. They were telling the local populations and all of the real estate from the manuals from the CDC and the WHO, from the billboards around, were all about don't touch, in some cases, your subsistence food. But after this came out, we started to see a real sea change shift where it was a lot about social distancing and hand washing. And this is sort of, kinds of places where I get excited about genomic surveillance data of how it can inform different components of the response. But ultimately, it was stopped and it could have been far worse, but it did go on to be a major tragedy with over 30,000 confirmed cases, over 11,000 confirmed deaths, likely still to be an underestimate. One of the great tragedies of it was also that the individuals that are always at most risk, and so I'm always delighted to speak to individuals in the, well, with an MD myself, I'm a researcher, but I do have care deeply about medicine and honor those who are on the front lines, because they are the heroes that take the most risk. And indeed, we lost many nurses and physicians in this outbreak, including Dr. Khan, who was the head of the ward, who passed away while caring for caring for actually another healthcare worker at the hospital, became infected and then passed away. And so I talk about this a lot as a tragedy, but on a precipice of a cataclysm. The same week that I learned about Dr. Khan's infection and passing, Dr. Christian Happy, my longtime collaborator of now 20 years, called me after leaving the lab, having diagnosed the first case of Ebola in Lagos, Nigeria, a city of over 10 million. And he did the test at Redeemers University, which is on the grounds of Redemption Church, where Christian and I are sitting here. And when he left me that night on the call, or that morning on the call, he had to get out quickly because they were preparing for First Friday. And this is the scene on the steps that we were sitting, just about 100 yards away from his lab in the same encampment. And I remember him telling me about the tanks coming in because this was a major target of Boko Haram, as one of the largest churches in the world. This church at the time sat 1 million and now seats 3 million. It's a sight to behold. But Christian and I have always been motivated by the thought that came to our head, which is, what if a single case of Ebola was to move to this crowd? And what if the index case did not want to be identified? All of this sort of points to the fact that, again, taking you back here, all of this is eerily familiar. All of the things I'm talking about, we all know. But at the time, it was sort of unusual to talk about. But fundamentally, we recognize that these viruses are here to stay, and potentially we're in a place of graver threat, as that the world is, there can be bad actors in the system. And so at the time, we started really laying out our proposal of what was needed. It's very simple and very intuitive, but it's the idea that we need to be able to detect pathogens wherever they occur. Using genome sequencing, we need to be able to read out the genomes and identify new pathogens and to characterize them, to then put those into place to get molecular diagnostic tests that could test for a wide variety of potential infections in hospitals. And also point of care tests that basically get us to the point where individuals could be tested in remote villages or in homes, so that no one ever has to come to the hospital to be tested for a deadly pathogen in the middle of a pandemic. We then need to be able to connect that information in real time, because ultimately, while all of modern medicine can use more data, infectious diseases is particularly dependent on it because the network matters. The fact is, these are communicable diseases. Within that context, all of this information informs information locally. And we don't want to just connect the different hubs, the different clinical hubs. We want to connect every individual actor. How do we bring information technology to get all best information at all times? And then we want to empower every actor in the system. We want to be able to feed back the data that each individual, each frontline worker, each public health expert needs in real time, that then incentivizes them to give the best data back. And we put that all together as we proposed creating Christian Happy and me and our teams put together this all in a thing that sort of describes all of the different components that are needed. And we're funded and supported by the Audacious project in February of 2020. Sort of, we were chosen in 2019 and got the final award in February 2020, just in time to sort of get into this. But even then now feels like audacious was not nearly audacious enough to fight what we were going to contend with. I'm just going to tell you about these different areas and some of the work that we're doing. I will apologize that I can't cover all the different things, and also that sort of as we're doing it, some things are fresh off the presses, some things are sort of trying to figure out how to get in to basically share data and act. And I think you guys talked about at the beginning of this how important it is to communicate all of this and how important medical rounds are. And so I'll do my best here. So from the genome sequencing standpoint, we talked about this. I talked about this a little bit before in the context of Ebola, but those kinds of things that this data can share, it can tell us about those transmission patterns. And just as we learned about Ebola spreading from human to human, early in the outbreak, my team and the Broad Institute worked closely with the Mass Department of Health and Mass General Hospital to characterize a conference in February of 2020, an international biotech conference that went on to have a very well-documented outbreak. And we were able to show, in fact, that this outbreak likely went on to impact. We see the mutations in that lineage showing up in hundreds of thousands of cases downstream, telling us kind of this effect of the super spreader event, and also really, seeing that almost a clonal expansion of a virus, suggesting that either one or a very small few individuals are spreading this virus. We were able to actually compare that also to other outbreaks that were happening. One of the things that we found was that same lineage did go on to see hundreds of thousands of cases, including many downstream super spreader events, including a number at different homeless shelters, reminding us that the choices we make as the privilege, being able to go to these conferences, will always disproportionately impact the most vulnerable amongst us. And so we don't really understand how they got into the homeless community, but it's a reminder. And of course, this conference did what was expected at the time and did a great job of documenting it. But it's for us to know now as we go through these different cycles. And just as we saw those novel mutations occur in Ebola, Jeremy Lubin contacted me in February of 2020 when he saw D614G mutation that caught his attention and sort of said, this looks like A82V all over again. And we began to investigate and showed that the mutation does increase infectivity and change the likely sort of protein conformation. And now we see that indeed, we see it throughout all the cases that we see. So in both cases, these mutations rose to fixation. And that became the beginning of many variants of concern. During a mumps outbreak that also happened, locally first on Harvard's campus is how I got attention to it, but then throughout the Massachusetts colleges, we became really interested in this idea of how the genomics can really inform transmission mapping. This basically outbreak started where we saw it on sports teams that moved into the dining hall, that moved into the health services where nurses were impacted, was found at a commencement event, that went on to actually, we found through genomic data that it went on to seed an outbreak in East Boston, where 3 individuals in that outbreak were affiliates of Harvard. But one of the kind of overarching things that we found was that there's a tremendous amount of contact tracing that was done during this outbreak, with really exceptional work by the Department of Health, as well as the different colleges. And they were able to identify a number of transmission events in this very fast-moving outbreak. But when we looked at this data, what we found was, and we overlaid the genomic sequencing data. Oops. What we found was that for all of the instances where we had an epidemiological link, it was confirmed by genomic sequencing. So traditional epi is quite accurate, but it missed so many likely transmissions. And so we understood that it was underpowered. So it kind of pointed us to the fact that we needed to bring genomic sequencing data to bear, but also that we really needed to figure out how to bring contact tracing into the information age. And we also, at the same time, really began working on building so Christian and I launched in 2013, actually just on the eve of the Ebola outbreak. We seem to have these big launch points just on the eve of a major outbreak. But we launched the African Center for Excellence of Genomics of Infectious Disease. Here's the building built by Mass Design and what's sort of going on currently in the building that we're about to move into, and the building that it now exists. But we, through ASCID, we established diagnostics in February 2020 in Nigeria, Sierra Leone, and Senegal, well ahead of any US hospital. We're running about 1,000 diagnostics a week and serving 30 million people. We've done sequencing for 14 different African countries. Actually, I think it's now almost 1,000 a day, I should say. And again, always trying to update in real time. But we've also been working on thinking about how to enhance diagnostics. So in addition to setting up these diagnostics in Nigeria, we also supported the Mass General Hospital and other hospitals in the Boston area to set up diagnostics. We scaled up our sequencing capacity here back in the Boston area, and developed a paper that just actually came out. It's in press as of yesterday. As of yesterday, it's actually published in Nature Microbiology, a technique to add genomics barcodes to the sample so that we can track the about 2,000 samples that we are sequencing each day at the Broad Institute, and also have rolled this out to a number of different sites in the New England area. And we've developed cloud-based analytics to allow people to do their own analysis or do other analysis, supporting other groups like the Next Strain platform, as well as our own, to be able to analyze this data in real time. We've also been doing, so we continue to, like many people, study these mutations, but to us, it was always clear that these mutations are popping up again and again. And we really wanted to focus on how do we pick these up in real time. Through my work in human genetics, became really interested in non-coding variants and other kind of variants off the beaten track. We had during the, not presented here, but for Ebola, we actually found a number of mutations in the NP protein that also looked like it was under selection, where we found biological effect. And so we wanted to say, how could you really look at this in on a global scale to say, what can we, rather than looking by eye like we've been doing, can we actually develop systems to do this in a large scale? And my former postdoc, Jacob Lemieux, who's now on faculty at MGH, has been leading this effort with Fritz Obermeier, who developed Pyro. Essentially, it's a Bayesian hierarchical model that infers relative transmissibility of all viral lineages across geographic regions. And importantly, also, we can use Pyro as a powerful way to analyze now over 5 million genomes and to look for evidence of convergent evolution to point to which mutations actually matter. And we find a lot of spike mutations you might expect, but many non-spike mutations like in the nucleocapsid and non-structural proteins. Just more recently, just kind of fresh off the presses, it's on medRxiv, but in press at Cell and coming out in the next days. We also investigated, you might remember when things sort of quieted down in the summer of 2021, and then suddenly Delta came on the scene. And there's a particular outbreak that caught national attention and changed the mask mandate policies, where there's an outbreak of Delta variant with similar viral loads in vaccinated and unvaccinated individuals. And we since went on to really use the genomic data to begin to try to understand transmission events and to see whether or not there were transmission events that began with vaccinated individuals. That is, can vaccinated individuals become infected? It appears yes. Can they actually infect others? And so we're looking to that and looking at the epi data. There was evidence that there was there are basically two potential links between fully vaccinated individuals. When we brought in the genomic data, we found many, many more of these links, likely plausible links. We confirmed the two that we had seen, and then also went on to find many more plausible ones. And then we basically cycled back. We kind of used that data to then say, could we now go back and really do some deeper dives of contact tracing to see if we can find more? And this is basically what now after genomic data was then confirmed by contact tracing or like a likely confirmation. And so there are many more links that the genomics probably points to, and many additional links that the genomics helped then get the specific contact tracing data to share. And all of that other stuff in the genomic data that isn't validated is a hypothesis and a lead to be followed. And the faster we can do this genomic work in real time, the faster we can actually follow these leads and see whether or not there's cryptic transmission we're not identifying. But ultimately, this gave pretty definitive evidence that we are seeing spread from vaccinated individuals. So that's the sequencing data, but one of the important things about the sequencing data is it can form diagnostics. And while there are a lot of really exciting diagnostics out there, I've kind of put my focus on CRISPR-based diagnostics. And as we all know, CRISPR has transformed modern medicine and has so many different utilities. But when it was first discovered in nature as a bacteria's immune system to viruses, it's able to essentially, these RNA the generated RNA are able to go and circulate, identify particular viral sequence, and then in doing so, pull down a Cas enzyme signals a cut. And in particular, enzyme Cas13 at the time I was looking at, now Cas12 has these properties. What's interesting about it is there's something called collateral effect, which is not, which could become problematic if you're thinking about therapies, but it's really interesting when thinking about diagnostics. Essentially, we know that CRISPR has these very precise cuts, and that's where it's really kind of tremendous about this ability to be these like molecular scissors that are very, very specific. But in this case, once the that very specific cut gets made and that signal starts, you start getting more permissive cutting, essentially what we call collateral effect. And you could use that collateral effect by attaching some sort of a visual readout to a quencher that then would be cut if this sort of permissive cutting happens, to then be used to create some sort of a visual readout we could see. And this is the work, again, that where I have conflicts of interest. So just want to point you to paying attention to that while we go. But I'll tell you, so I came into it during the Zika outbreak. We were doing all of the sequencing data. So we were a lab that was a lot of bioengineers and sort of bioinformaticians studying viruses, but by collecting a lot of samples from them, sequencing the genomes and analyzing them. And we were seeing that in the case of Zika, we were interested and began working on Zika because we were kind of called into it. A lot of groups around the world were, and particularly in the Americas, were not able to sequence the virus, and they asked us to help. And we started to see the problem. Zika is a virus that is a very low titer. Unlike Ebola, it's like very low concentration in different clinical specimens and doesn't stay around for a very long time. So you have to find a diagnostic that is really sensitive and can kind of be done quickly. But through this process, we ended up sequencing over 110 samples from 10 different countries. And for every country where we had enough data to be able to say something about the origin, to date back the origin of the virus in that country, the likely origin of that virus came many, many months and now likely well over a year before the first confirmed case in that country. And so it told us that we were missing this. We were giving viruses a long time to get started, and that we needed better diagnostics. And at that point, Feng Zhang and Jim Collins, my colleagues at the Broad Institute and MIT, were working on developing a CRISPR-based diagnostic, and we partnered to test this on Zika virus. And my lab then kind of went further to really try to move this to point of care. We were working on different components of allowing us to start with non-invasive specimens like urine and saliva, to use methods that don't require machinery, and fundamentally got it to the point now where we have a paper on the bioRxiv that can go end to end with no equipment, that can work at room temperature, that is lyophilized from end to end, that we are able to process in countries like Nigeria, to do things at massive throughput, to be able to test hundreds of different patient samples, hundreds of different viruses simultaneously, and to develop machine learning models to better design these assays, to essentially use all of the data we can generate through the massive throughput to better feed the model and test lots of different guides and identify the ones that are the best. And so from the sort of achieving the goals, just showing a few data points, this enzyme, the Cas system is incredibly sensitive to attomolar sensitivity. It can be fully field deployable, working on urine saliva. It can distinguish different subtypes and variants, different specific mutations, and we can develop these assays very, very quickly. This is 6 days to developing the assay for our Zika virus, but now in 1 day, we can design new guides for specific viruses or specific mutants of viruses. And we've been able to put this into the field, and in Nigeria, lyophilized samples, sent them 6 weeks later and had them working in Nigeria. From the component of our system for doing high throughput diagnostics, it's called Carmen. And this is really a combinatorial system that we're using that uses microfluidics and other forms of miniaturization to allow the ability to do this. And this works very well because of the fact that CRISPR is so sensitive and specific, and can be done where you can combine a number of different, essentially, a lot of this can all work in the same conditions, allowing you to do a lot of things in this kind of multiplex fashion. And ultimately, we can show this is 169 viruses known to infect humans, where there was enough genomic data to design guides against 169 targets. And we show that it's exquisite sensitivity and specificity. And in the blue here with patient samples, always sort of finding the virus you expect, and then sometimes finding other viruses that are caught by next generation sequencing, like anellovirus known to infect blood. And we could also distinguish these very unique subtypes. I have some data that I couldn't quite get in because it's really fresh off the press, but we did also move this into a commercial system that we could implement at the Mass General Hospital as well as at the Broad Institute, and now have this working both at Princeton University, where my former postdoc leading this work, Cameron Miravold, is, and our colleagues at the CDC are running. But this is using the Fluidime system, we were able to incorporate this and make it work, test about 1,000 samples and show that you could test for 21 different viruses very, very well. And we went all the way through the FDA submission process with them. And basically at that point got locked out as an EUA, but encouraged to submit a full submission. And the data that it's fresh off the presses, I didn't get a chance to get to, like literally fresh off the presses, and I was talking about it last night, is that we're now able to use this to, within a matter of hours, identify Omicron cases. And so we had a number of outbreaks that were happening on different college campuses. People are panicking, and we pulled out this 22 variant panel that can uniquely identify all of the different lineages. And we're able to show that, this system can really work to doing this type of triage in real time. So that's a little bit about the different ways that we're thinking about detecting different viruses. We've also been working to connect all that information, working with our partners, Demagi and Fathom. Over the last decade or so, we have built a lot of different systems and built upon systems they've created to do sample tracking, contact tracing, different visualizations and citizen reporting, and really bring in that diagnostic and genomic data. And we built something called Viral NGS that has been running since 2014 in Nigeria, Sierra Leone, Senegal, and Liberia, and now is being used across the world to do metagenomic sequencing analysis. We've created Banyan with our partners at Fathom, which allows you to, a lot of the different systems that show phylogenies of viruses can only take a few thousand viruses at a time. You can't even analyze your whole data set, but we've worked to really improve that backend infrastructure so that you can actually sweep through looking at all 5 million genomes simultaneously, and then hone in on the different countries, regions, sample sets that you want to look at. We're about to release that, are working through some permissions in doing so. There's short video that shows you like kind of that exploration of that data and clicking on different groups that you want to look at. But we also, this is a little bit of an, there's a number of different features that are not shown here, but it also allows you to look at lots of different, all the different mutations and sort of scan and look at the different mutations and also look at different geographies. I think it'll only be showing part of those right now in this video that I should have within that context. I think you'll sort of see that you can also scan through and see all the different mutations and begin to investigate them. We've also been, so since 2016, during mumps, we became really passionate about how information technology can help. Essentially what we started designing, the pitch I was giving and was using, I was building it at and for Harvard, was the Facebook app for outbreaks that also needed to start with a close-knit community where you could get buy-in, where you could actually give people back actionable information. And so this is a way that people could give in their symptoms and kind of track their symptoms. Like you get a cough and you're like, what's going on here? Well, actually like 6 people in your intro to genetics class have these symptoms. Actually, one of them has mumps. You might want to get tested for mumps, but also gives a back-end dashboard for the universities and communities to see. And so you can see here, this is just showing like, there's a cool visualization I can show if we have time, but essentially this is all moving in this sort of multimedia fashion, but that where you can sort of click on a campus, then click on a dorm, and then identify, and then click on a person and see all of their contacts and start doing really this the epi work that we see our colleagues doing all the time on, by hand in Excel on the back of an envelope, giving them the tools to really make public health local, and feed up that data to regional, state, regional, national. And also to integrate that genomic sequencing data. So this is data from anonymized data from one of the colleges that we work with, but you can sort of see that you can click, there's all these different frames that you're looking at, like the wastewater and symptoms and the dorms and a kind of global map, but you can also click and see what's happening at the sequence level and identify different clusters and see whether or not individuals are on shared teams or what, basically doing all those investigations all under one roof. From an Empower standpoint, I just want to point to the fact that with ACEGID, we've now trained over 1,200 individuals from around throughout West Africa, as well as individuals in Southeast Asia. We've worked with the CDC to give on-site training. Oops, I am running out of power. Goodness gracious. Okay, I am not near a cord. I moved up to a specific location. Let's see how this goes. I'll just try to stop this quickly and then let me see where I'm at with power. I've got 12%. Okay, we're good. I'm gonna just rock this as fast as I can and then go find. But essentially we worked with the CDC to train a lot of their different staff. This is these are some of our trainings. We did these 7-week trainings at the Broad Institute over many, many summers. We also developed this, app that spreads. So we were working with Todd Brown at Sarasota Military Prep Academy, helping him to develop an education curriculum for his students. 7th and 8th grade teacher who came to me in 2015 had me engage with the students and started talking to me about how we could teach kids about outbreaks. So I was advising him, and one of the things that we were doing, it was basically simulating an outbreak. And the problem with that was just, how do you spread it? We tried spreading it with stickers and lollipops and it didn't work well. And at the same time, we were thinking about those apps to track outbreaks and how Bluetooth could come into handy. And so Andres Calubri, a postdoc in my group, built an app that spreads a virtual virus via Bluetooth. This is the old frames. They're much more nice now, and you can see them and download them on the app stores. But essentially can model every aspect of an outbreak and gives you really, both teaches outbreak science very well, but also gives you really interesting data. And so this is a simulation that we ran where those contacts in the middle that you're seeing, those are real interactions between students on the 358 students on a college campus we're working with. It's Halloween weekend, these kids partied, and by early November, there was a real outbreak on campus, and we got to see what it would look like. So this is a virtual virus spreading, but you can really look and see all of these interactions, and the students really engaged with this because they could be like, oh, this is how it looks like, because you're trying to tell people this virus could spread, but they don't really know, you can't see it. So here's really getting that ground truth data to engage with. And also, if you click on this, you get all sorts of different data points, but you can get how many contacts each person has, how many secondary contacts they have, that person that has only 3 friends, but their 3 friends have 250 friends. We found some of those. So all of that kind of tells us things. And we have a textbook that is coming out, is actually being piloted in the winter, starting in January, for a statewide course for the Department of Education of Louisiana for all 11th and 12th graders. And we have a crash course that goes with it that just wrapped. And then finally on Overcome, I just want to say that there's a lot of other technologies I'm actually not getting a chance to speak about here, but the kind of work we do informs therapies. We're building CRISPR-based antiviral therapies. We're reading out the host and viral expression in single cells. We are identifying what viruses are presented to our own immune cells. And I'll talk more about that tomorrow at the ID and Immunology lecture. We're also, in the human side, we've been doing a lot of work developing massively parallel reporter assay to detect human regulatory variants, both throughout the genome and in 3' UTR variants. And we just have a paper that came out showing that we could use our expertise in viruses to essentially do gain of function work in AAVs, but allowing them and training them to target very specific tissues and understanding how that works. Oops, that's the same slide. But so with that, I just want to thank Christian Happy, my colleague, the many amazing partners that I've had, including Kate Rubens, who brought me into, a lot into this world, who's a friend of David and I, who's a master astronaut. But Christian, like I said, Christian being the major colleague, but these individuals all represent huge teams that we work with, all of our different funders and supporters. My lab, who's about to release their biggest publication of the year, our annual holiday card, which is coming to you very shortly. I'm actually going to move because I don't want to run out. And then all of our international collaborations that make this work possible. And finally, the patients who allow us to do this, the sort of citizens around the world who allow us to do this work, who trust us to do it well, and who inspire us and motivate us. And I think with that, I will close and take questions and find power.
Thanks, Dr. Sabeti. Sorry you're running around looking for.
Oh, I totally forgot about that. That's a rookie mistake. So
I know from the emails you've been working a minimum 12+ hour days and then being with us to present. So having the time to find power cords and stuff, I'm surprised you have time to do anything else. So thanks so much. We'll make sure we close this right at 9 in case you're running low on power. I want to just remind everybody, there's thanks, Dr. Lubitz, for your question. Any other questions, please throw them up. I want to turn it over to Dr. Singh, who has a comment as well.
Hi, Pardis. This is Upi. Can you open my video, Errol? I can't undo it.
It has to come from you. I can't actually.
No,
no worries.
Okay. No, it's okay. I can't do it. So it was fantastic. And like, a tour de force, I guess from your perspective at both the front lines as well as the 30,000-foot view, we've learned so much, right? From the last year and a half, 2 years. Where do you think that and you've shown us ways that we can engage the scientific community, the clinical community, as well as patients themselves. Where do you think are the next issues that really have to be addressed before the next pandemic, right? Whether it's a local versus, international pandemic, as this has been, like, where would you focus your efforts? Where should governments or we focus our efforts?
I think there's a lot of different places, but I would say, oh, hi, and good to see you now. So I would say, like, where we're at right now is, the technologies exist and they're in place and they're like, this is not rocket science or we've become a rocket, or it is rocket science and we're there, but essentially, there are diagnostic tests that can be used at point of care. There are sequencing approaches that can really characterize these viruses all around. We need to, and there's information technologies that could be used in a really powerful way, but they have to be supported. I mean, the funny thing is it's amazing, this like Carmen assay that we have, we can do, thousands of patient samples on numerous viruses. And we are still trying to get support for it. And I'm just one of many, right? This is a story as old as time where it's like, that basically the FDA said you need a 510K. And I'm like, well, that takes money and I don't know where to get that from, right? But like that, there isn't that kind of support. We're all hearing money is coming, but it's very few pockets. So I think more support needs to happen to bring that to bear. It has to be distributed in a transparent and merit-based way, because there's a lot of that kind of stuff where it's like, where is it going? How do we actually tap into this? So all of that's there, but I would say, like all of those different pieces of getting genomic surveillance, getting to a position you can immediately roll those out to diagnostics and vaccines and therapies, being coordinated, all need to happen. But the kind of the broader thing we see is that as much as the technology, it's the culture. So I wrote a book with journalist Lara Salahi called Outbreak Culture, about the fact that, outbreaks really prey upon us. They're a crucible where you have this like convergence of an insidious, deadly threat. And we saw this and, we see this time and time again, but sort of playing out here where so much of our clashes this year have been cultural and so much, understanding is not happening. And so ultimately there's something that needs to truly happen and lean in to make this more, there's a concept called organizational justice, where things have to be equitable. They have to be transparent. They have to be merit-based. You have to communicate in those real ways. That's where we need to get to. And until then, these viruses are going to expose and exploit the cracks in that justice that we have as a society. And so there's a lot of work to be done, but there's a path. It's just about committing to pursue it.
Great. I think we also have some questions, Errol.
Yeah. So a couple questions here if we can get to. Terrific presentation. Why do you think healthcare facilities have not yet adopted prospective molecular epi for outbreak detection, infection prevention outside of research? How can we get there? Understanding chains of transmission of respiratory viruses and bacteria could help us target our effort as it did understanding the dynamics with Ebola.
Yeah, I mean, it's the great question. I think that's actually that's one of the things that motivates us, right? Is I join a panel all the time with epidemiologists, state and local epidemiologists around the country, as well as work with a lot of different local providers. I mean, it's because we're all understaffed, we're under supported, we are reactive, because we have to be, and no one's going to stop and learn how to use these tools in the middle of what they have to do in their day job. So that's actually, that system is called Lookout, the one that shows you all the epi data and then combines the genomic data. That's a huge passion project of mine, because really, in order to make that happen, you just have to give people tools that are easy and accessible that allow them to do that work. And so we're just right now just working with individual partners that can kind of come to the table with us and help us keep piloting it. And we have more and more. We've worked with Colorado Mesa University, UCF, SpaceX. We've a number of different partners trying to really test different components of these systems. But we are, looking to roll that out and expand it, and also work with different. We're starting a project with the Mass Department of Health, but it hasn't been the right time to try to bring those things on, because everyone's like, I can't deal with a new system right now. So we're working with a couple of partners to build it.
Dr. Sabeti, maybe one more question here before we close it up. Dr. Lupitz is asking about transmission, particularly given Omicron. Do you have an opinion about whether multi-mutated variants such as Omicron result from serial transmission on multiple hosts with added mutations to each host, or a single or singly from an immunocompromised host who can't eliminate the virus because of the weakened immune system?
Yeah, and it's both cases. We see them in both cases, and we've seen a lot of these like long. It doesn't have to necessarily be in a weakened immune system. Some people are actually just natural reservoirs. We see this in a lot of other viruses. Like all of these concepts, like asymptomatic infection, or or like long disease, or the sort of like long-term infections. We've seen them. We see them in Ebola. We see them in a lot of other viruses. So they can happen, and they can definitely be a breeding ground for change. But we also are knowing that we are missing a lot of cases, and it tells you that in all these places where we don't have any visibility, it's places where new mutations can breed. And so both are very, very likely. And the more surveillance we have, the less likely it is just through general transmissions. And then the more we really pay attention to those immunocompromised or supported, or do additional tests to see if anybody's keeping the virus for longer, the more we'll be able to stop the other one. So like in a larger world, we would have surveillance everywhere, and then everyone would have some test that clears them of the virus later downstream, rather than say we assume that it's gone, because it's usually gone, but not always.
Great. Dr. Sabeti, thank you so much. We're just at 9. Want to be respectful of your time. Again, want to thank you for being with us. And I hear you're hanging out. The Lucky Infectious Disease Division is going to have you as well speaking tomorrow. Thank you for all the panelists again. Just want to echo Dr. Harrington said earlier. Thank you to everybody who's been part of these medical grand rounds and everything we do here in this community. And I hope everybody has a great holiday break. We'll see you again at the beginning of the new year with Dr. Fearon kicking this off the first week of 2022.